Okay, perfect. So, hello again. In this second talk, I will talk a little bit about the specific technique, CD spectroscopy, and especially how to analyze protein folding or protein fold state using CD spectroscopy. So first of all, I want to introduce to you to the principle of circular dichroism, because a lot of people know CD and use CD, but the general principle behind it is always a mystery. So I really want to go into what is circular dichroism to begin with. Then we have a look at CD spectroscopy on proteins, how it is measured, and how CD spectra are properly displayed or compared. And once you want to compare CD spectra to each other, you run into an issue, and I will elaborate on that, because you can, in principle, do a lot of things, but don't overdo it. Yeah, do it reasonably. So first of all, what is circular dichroism? It's a combination of two words. We have circular, coming from circular polarized light, and dichroism from two kinds of light that are treated differently. So we have something that treats two kinds of light differently. So it's a material property. And if we talk about light, then typically you envision light as an electromagnetic wave. You have an electric field that's oscillating, and you have a magnetic field. So in the next slides, you don't see the magnetic field at all. So just imagine there is a magnetic field as well, which is perpendicular. But things get too messy if you see both of them. So what you, I hope you can see that. What you have is, for instance, a horizontal polarized light. That would mean that you have an electric field that is oscillating in a horizontal plane. What you could also have is vertical polarized light, so light that is oscillating the electrical field in a vertical plane. So horizontal, vertical polarized light. And I think this is a concept that you all know from basic physics courses at one point. You have these polarization filters. And what you can do now is you can mix them. You have horizontal and vertical polarized light at the same time. And when they come together and there is no phase shift, so which means the 0 points meet each other and the maxima meet each other at every time point, you just do a mathematical superposition. And what you end up with is when the green and the red one, you always add them up. You come up with a vector, which is the blue one. And in this case, the resulting vector is just a linear polarized light again, but 45 degrees. Okay, so what you can do now is, instead of having them without phase shift, you can introduce a 90 degrees phase shift. That means that whenever the vertical polarized light, the green one is 0, here you see it, the red one is maximum. And on the other hand, when the red one is 0, the green one is maximum. And you do the same principle. Again, you just do a vector addition of the green and the red one. And what you end up with is now the blue vector. And interestingly, the blue vector now has a constant amplitude, and it's rotating. Yeah, so you have no oscillation through 0, but what you have is you have a constant amplitude rotating, and in this case, it's called left-handed circular polarized light. Left-handed because if you look at the light coming towards you, it's like the left hand, you look at the thumb, and the electric field is rotating in this direction. So that's left-handed circular polarized light. And you get exactly the same thing with a minus 90 degrees phase shift, so now the other one is coming earlier. And what you will end up with is right-handed circular polarized light. Again, you have an electric field with a constant amplitude, but it's rotating counterclockwise. So that's left-handed and right-handed circular polarized light. And now, we think always light of either polarization, like horizontal, vertical polarized, but actually, this is a mixture, or it's not well defined. If you look at light that comes to your eyes, you cannot really define what it is. You can say it's a mixture of everything. And actually, what we see here, those are just mathematical expressions, because nobody knows what light really is. But you can nicely describe it with these equations. So what you can think of is, you have the left and right-handed circular polarized light. But what you can think of now is you have your vertical polarized light, the blue one. You can think of it as a mixture of left and right-handed circular polarized light at the same time. Because what you get is when you, again, sum up the left-handed and the right-handed with no phase shift, you do vector addition, and what will happen is that the horizontal parts will always cancel out, and you just add up the vertical parts. So essentially, by adding up left and right-handed circular polarized light with no phase shift, you get vertical polarized light, or with a certain phase shift, you also get horizontal polarized light. But all these is just mathematical expressions. So there is no difference between vertical polarized light or a mixture of left and right-handed. It's just a different explanation of the same thing. So we can think of vertical polarized light actually as a mixture of left and right-handed circular polarized light. And what you do now is you just do spectroscopy. You measure absorbance of light. You have a cuvette, you have a sample, you shine light on it. In this case, we have left-handed circular polarized light. The light will be absorbed. After the cuvette, you will have less light. You just have an absorption experiment, similar to ordinary spectroscopy. And now something strange is happening. You have, again, your sample. Just look at the blue lines first. You just shine vertical polarized light onto your sample. And when you look at the light that comes out after your cuvette, after your sample, you see it's not vertical polarized anymore, it's elliptic. The blue light is now not only vertical polarized, it also has a horizontal part. So how can that be explained? So people say now, whatever is in there, it has some ellipticity. It's optically active, it changes the light. And how can we explain that is, we can just look at now the left and right-handed circular polarized light, the green and the red one. And they have the same amplitude when they come in. Now they are absorbed. And if you now think that the green one is absorbed a little bit more than the red one, like the green one is smaller than the red one, you do vector addition of the green and the red one again. And what you can come up with is the blue one. That means that this sample, whatever is in here, absorbs left-handed circular polarized light stronger than right-handed circular polarized light, which means the sample that's in there shows circular dichroism. It distinguishes between left and right-handed polarized light. That's the definition of circular dichroism. It's a property of the material that you have in here. And in CT spectroscopy, you look exactly at this phenomenon. And what we look at is, when can we see that? So essentially, since the molecule has to distinguish between a left and right-handed polarized light, the only way that is possible is you have a chiral molecule. Chirality of the molecule will distinguish between left and right-handed polarized light. If you have a symmetric molecule, there will be no circular dichroism. And lucky us, proteins are chiral. That's why you can do CD spectroscopy on proteins. You have the C-alpha, which is intrinsically chiral. It absorbs below 200 nanometers, but you also have peptide structures at around 190, 200 nanometers, the peptide bond, and this also shows chirality. Because you have the alpha helix structure, which means the peptide bond has a certain chiral features, and also the beta sheets, they have certain chiral features. Yeah? So the protein will show circular dichroism because it has some chirality inherent either by the C-alpha itself or by the secondary structure that it contains. So what do we actually measure? We measure now the absorbance of the left-handed circular polarized light. We subtract the absorbance of the right-handed polarized light, and this is called delta A. So it's the difference in absorption. And this difference, as I showed you before, will make this ellipticity, but in reality, this difference is really, really tiny. So the difference between left and right-handed circular polarized light absorption is just 10 to the power of -4 compared to the ordinary absorption. So it's really a really tiny, tiny effect. So that you need specialized instrumentation to be able to measure these effects. And for historical reasons, you don't display delta A. Sometimes in the more recent publications, you see delta A, the difference in absorption. But in the more ancient publication, you look at the so-called ellipticity. And the ellipticity is just as I showed you, how elliptical is now the light after going through the sample. And those two are just related through a number. And what you typically do is, if you look at the CD spectrum, you don't look at the absolute CD spectrum, but you want to normalize it. You normalize it by the unit of absorption. And in this case, the unit of absorption, it depends, you normalize by the concentration. So if you have a certain concentration of your protein in there, you normalize by the path length, obviously, if you use different path lengths, like in spectroscopy. But in this case, what we typically also do is we normalize by the number of amino acids -1. So you normalize per peptide bond, because that's the unit of absorption in CD spectroscopy. And if you normalize your ellipticity like that, then you get the so-called mean residue ellipticity. So per peptide bond, you get this spectral feature. And this is what you typically show, and this is what you typically compared. And this is a CD spectrum of proteins, and if you look at the blue CD spectrum, that's what you see. You have quite high CD spectroscopy at around 190, 200 nanometer, and that's where the peptide bonds absorb, and there essentially you get secondary structure information. If you now go to this 280, essentially you have much, much, much less absorption there, therefore you have also much less circular dichroism there. So if you scale this effect here by a factor of 200, you also see circular dichroism coming from tryptophanes, tyrosines, and things like that. So you have essentially what people often refer to as tertiary structure information. And applied photophysics also likes to look at higher wavelengths, and you can have metallo-organic complex or other things. So essentially, as long as you have chiral entities that absorb, you can do circular dichroism spectroscopy in them. What we typically do in protein science is we work on far UV spectroscopy, so we really look at the region of about 180 nanometers to 260 nanometers. That's where the peptide bond absorbs. And the interesting thing here is now, maybe you've seen these slides over and over again, that different secondary structures like alpha helix, beta sheet, coil, turns, or whatsoever, they give you different spectral features. And this is now where essentially the structural information comes in. So CD spectroscopy is something that tells you about the structural properties of your protein. The other thing is CD spectroscopy is essentially different for each and every protein, because every protein has its individual CD spectrum. Again, here this is how it's normalized. So how do we measure it? This is the instruments that we have in Vienna. Here in Brno, they have a JASCO instrument that's applied photophysics. There are different manufacturers, obviously. But in principle, it's set up in a similar way as an ordinary spectrometer. You have a lamp, you have a monochromator, and now what you just have is you separate between horizontal and vertical polarized light, and you use a feature called photoelectric modulator to generate left and right-handed polarized light after each other. You send those through your sample, and you detect it. And then the software automatically calculates the difference between the left and right-handed absorption. Oops, wrong direction. OK, so what do we measure? We measure mainly on proteins. As I said before, each protein has its own spectrum. So essentially, the CD spectrum is something like a fingerprint. And this is a very important thing, because if you want to do batch-to-batch comparison, you just look at the CD spectrum. Does it look the same the next time? Then you know it's the same protein. And if you have any structural deterioration or any structural changes, they will be reflected in the CD spectrum. Another thing is you want to look at these structural changes. And the nice thing is that this CD spectrum is such a powerful method, or sort of trustable method, that actually this comparison is an FDA-approved method. So really, pharmaceutical companies use these CD spectroscopy for batch-to-batch comparison. And this is fully approved. Another thing that you can do is you can look at thermal stability. You measure the CD spectrum and you heat it up while you're measuring, or just sit on one wavelength, however. You can also do kinetics. You can follow actually the folding or unfolding of your protein. You can measure interaction. If you have small molecules or other interaction partner that upon interaction change the structure, then this is something that you can follow by CD spectroscopy. You can also get information about secondary structure, but I will show you that it's not so straightforward, and it's, I would say, overused by people. Another thing that you can do is obviously also CD spectroscopy in DNA and RNA. So you'll just look at 260 nanometers, and you have different chiral entities in DNA and RNA. I have absolutely no experience with that, to be honest, but I know people here in SATEC are doing CD spectroscopy on nucleotides. So what I did is show you now some samples that we recently measured. And one thing that you typically want to do is you have a wild-type protein, you make a mutant, and you want to make sure, does the mutant still has proper fold? Because for sure you make a one-point mutation, and the one-point mutation doesn't show binding. But is that because you mutated the binding site, or is it because you completely abolished the structure of the protein? And this is a nice thing that you can do with CD spectroscopy. You compare them. So another thing that you can do is you check the stability. Does the stability change if I add something? Does the stability change if I have a ligand or if I have a mutation? You can either thermally unfold, similar as David said before for thermofluor or protoplex. You can do that in CD as well. But you can also chemically unfolding using caltropes like urea or guanidinium chloride and just back calculate stabilities from that. And then again, secondary structure determination I will show you. And I will also show you a way how to quantify if the structural changes are significant or not, because I think that's one of the things that people really want to know. Is the difference significant or not? So first of all, that's a sample that we measured on campus, and I can't tell you which protein is it because it's not published yet, but what you have is you have a wild-type protein, which is the red one, and you have a mutant protein. And I got this sample from the customer, and they told me it's this concentration, and both of them have exactly the same concentration. I measure them, and it looks like this. So now, is that different or not? Or can you rely on that or not? So essentially, what you have to do is you have to normalize by the concentration. They have the same concentration. At least that's what they claim. So they look like that. But instead of using the concentration that people give me and the D that I have, you can use the path lengths of the cuvette. You can now apply Lambert-Beer's law. So instead of D times C, you can also have the absorption measured divided by the extinction coefficient. And this is a nice thing because protein extinction coefficients for 2 or 5 nanometers can readily be calculated from the amino acid sequence, from the primary sequence. And there is a nice tool here, and there's publications where you just paste your primary sequence, and the extinction coefficient for 2 or 5 nanometer is spit out. And the other nice thing is the spectral region that we're looking at is exactly where the 205 is. Here is 205. So we exactly measure actually the peptide bond. And you can use this extinction coefficient of your protein, and you measure the absorption at 205 at the same time, because your CD spectrometer is a spectrometer, so you also measure absorption there. So you can actually measure the absorption of your sample while it's in the cuvette, while you measure CD spectroscopy. And what I typically do is, or what we typically do, is instead of normalizing, it's back. Instead of normalizing with this equation, I normalize by the absorption that I measure in the same cuvette of the same sample that I measure CD with. And if I do that, suddenly the spectra look like that. And you see, the only difference that I had before was just the unreliability of the concentration that people think they have. You can even zoom in here a little bit. And you can, since we did replicates, you can see that's the 2 standard deviation range, and most of the spectrum is actually within that. So you can even say, maybe it's tiny differences, but okay, most of the people would say this is still similar. Okay. On the other hand, we also compared now the human, the Homo sapiens, and the Arabidopsis homologs. So it's the same protein, but in different species. And here you can nicely see that there are structural differences between them. There's also no sequence similarity, or few sequence similarity between that. But this is something that you can definitely see. There are differences in structures. One more time back to the concentration issue. I had 0.3 mg per mil lysozyme, and I just did a 1-to-1 and 1-to-1 dilution, and I measured this concentration with a very famous nanodrop that a lot of people maybe know. And then I normalized the spectra using the concentration that the nanodrop gave me. And the spectra look like that. This is the normalized spectra. And you can see that the variation between the spectra is almost by a factor of 2. Well, if you would use the same samples and normalize it with the CD spectrometer absorption method, they perfectly align on top of each other. So since you always look at normalized CD spectra, you have to know the concentration very accurately. If the concentration is 10 or 20% wrong, and you compare 2 spectra, then you only have 20% accuracy of comparing them. So keep that in mind. In this case, concentration is a big issue. And don't use nanodrop, use cuvettes. Usually, the concentration range that you work with, 1 centimeter cuvette is fine. We have 1 centimeter cuvettes that use only 70 microliters. The other thing you can do with CD spectroscopy is you can measure structural stability, for instance, melting or heating up. So I have here a fusion protein of GFP and GST, and you heat it up, and what you get now is you get the CD spectrum in this direction. So that's the millidegrees, that's the spectrum, as you nicely see, and then this is temperature. So it's a 3-dimensional diagram. And if you look at this direction, you just see the spectrum, how it looks like. And if you go in that direction, you see at a certain point here, the spectrum is going to change. And another point, the spectrum is going to change again. So what you get is you get two transitions. You get two midpoint of transitions. And you can also calculate the delta H for each of these transitions. And what is more important, you also get the spectra corresponding to the first species, the spectra corresponding to the species after the first transition, and you get the spectrum to the species corresponding after everything is unfolded. And now you also get structural information here, because if you look at this gray and the red curve, you can already say which of those two domains is unfolding first. Any guesses here? Yeah? So here you have nicely alpha helices, and alpha helices have this double negative peak, yeah? And this is gone after the first transition. So you can already say that this thing here is going to unfold at 51 degrees, while this thing here is going to unfold at around 80. Yeah? So there is already structural information in addition to thermal information. What people like to do, and what I was also very enthusiastic to begin with, is extracting secondary structure information from your CD spectrum. So you have your CD spectrum, that's the black one. And since I know how typical alpha helix, typical beta sheet spectra look like, there should be a possibility to extract that. And there is, and you can calculate actually from this spectrum how much alpha helix do I have and how much beta sheet do I have. But now the issue starts, because what you do is, and how you come up with that, is you compare your CD spectrum to a lot of different protein spectra. Now, there are algorithms that take a lot of protein spectra and compare it to yours. And you can use a lot of different sets of protein spectra data sheets. Yeah? And depending on which of the reference spectra sets you use, you get different results. The other thing is you can use different algorithms to extract secondary structure elements. And depending on which algorithm you use, you get different results. And last but not least, what we have is we have a concentration dependence in this normalization, as we saw before. If the concentration was not properly known, or if the concentration is 10% off, the overall spectrum will be bigger or smaller, and now also the alpha helix and beta sheet content will be off. Yeah? So there are a lot of issues, like accurate concentration measurements. The reference data sets are actually not accurate. How can you believe those reference data sets? How accurately was the concentration measured there? You can use different algorithms. So what I did till now is I just compared several algorithms and see if they have a common denominator. And one other issue that I just recently realized is that actually all the instrumentations have to be properly calibrated. And unfortunately, that's not the case. ARBRE-MOBIEU made a CD benchmark. Actually, we have a pre-benchmark, and we after that also did a full CD benchmark. I don't have any data on the full benchmark, but I was part of the pre-benchmark where we tested 8 different instruments in Europe, and Nicolas Kmel was heading this benchmark. And the idea was also to find actually a suitable calibration method, because the CD spectroscopy instruments are not very well or not properly calibrated. Up until now, the method of choice is using camphor sulfuronic acid, CSA, which is a chemical that shows this CD spectrum. And it has a single peak. And it's just positive. There is only one enantiomer. And actually, it absorbs somewhere at 290 nanometer. So that's not where you typically look at the protein. Typically look at protein at 200 nanometers. And you have only one enantiomer. Yeah? So applied photophysics is spending a lot of effort on establishing an optomechanical feature to calibrate the instrumentation. And I don't go into detail about that because they don't sell it. They just come to you and do it. But another option is what Nicolas Kmel was working on together with Jasko, and ARBRE was using this compound in the benchmark. This actually is using 2 pure chemical enantiomers. So you have this sodium cobalt EDDS in either RR or in SS conformation. So it's 2 pure conformers. And now the advantage is you get 2 CD spectra. First of all, you have several peaks of the CD spectra, and also you have it over a broad range of the spectrum. So you don't only have a single peak, but you have positive and negative peaks, and over a wide range. And now we had 8 different CD spectrometer, and everybody got exactly the same lysozyme sample. That's what it looked like. Also, everybody got the same 2 enantiomers, and that's what they look like. And when you, when you zoom in, now you see the big issues. Because if you look at pure right and left enantiomers, when they cross, that's where the 0 should be. So essentially, the interesting part is that where all these cross. And you see they're not only crossing some above, some below 0. So the calibration is not the same. The amplitudes are different. So you have amplitude calibration issues, you have shift issues, and also if you look at that, also the x-axis, so the wavelength was not properly calibrated. And this is now the issue that we're facing. You want to compare your CD spectrum to another CD spectrum or to a lot of other CD spectrum of other groups to extract secondary structure. And this is just very difficult. It's easy to compare relative changes. If you just have unfolding, you look at it, how does it look before and after? You add something, how does it change? This is something you can easily compare. But the moment you try to compare absolute spectra on different CD instruments, hell breaks loose. Because you want to make sure that the spectrum I measured here and the spectrum I measured there actually match. And the only way to do that is that you always also measure a calibrant at the same time and use the calibrant to correct for your CD spectrum. And this is something that people not readily do right now, but this is just in the process of approaching. And also an issue is accurate concentration measurement. So another thing is if you measure on the same instrument, you don't bother. Because it's the same instrument, you just make sure that the concentration is reliably measured. And what you usually want to know, is there a significant difference? I measure this one, I measure the mutant, is there a change? And how this can be done, applied photophysics approached this with a new method which they called weighted spectral differences. And I like this, and I guess you will also like it because it's tailored for biologists, because you get a single number. And you look at a single number and just see if this is nice or not. And so what you do is essentially you calculate the so-called weighted spectral differences. You look at the difference between your spectrum and the mutant spectrum. You weight it in a way like the parts that have higher are more weighted than others. You sum it up and you come up with a number. And this number is called weighted spectral difference. And now what you can get is these really nice plots. You have the weighted spectral difference for your reference, your weighted spectral difference if you do a dialysis, and the weighted spectral difference if you treat your sample with certain things, or if you add something, or if you change pH, or whatever. And if this weighted spectral difference shifts out of 2 standard deviations, you say, I have a significant structural change. You don't say it's more alpha helix, it's more beta, but at least you can say there is a significant difference. Or if you have a mutant and you want to say, okay, the mutant has still the same structure, you can say there is no significant structural differences. Yeah, with that I want to conclude, and hopefully I showed you what circular dichroism is to begin with, to get the grasp on that. What does the CD spectrum depend on at proteins, and what things you can do. Structure determination I made in brackets because it's not so easy, but you still can do it as fingerprinting. You look if your protein is the same. And if you want to compare CD spectra, make sure you do it properly. Thank you.